So today at the ISCBacademy, we're actually continuing on the topic of mutational signatures. Some of you may have seen yesterday's talk from Marcos, who also talked about the developments in this field, and I will continue along those lines. So what I want to talk about today is mutational processes and how they act on the cells in our body, and how this can lead to cancer or other diseases. I will be discussing some of the computational approaches that other people and I have taken in the field to study these mutational processes. And I will give you some examples from my work as well. So I will first start by giving quite a broad introduction into how these mutational processes look like, what they do to our cells, and how we can identify them computationally. And then I will talk about some applications of studying mutational signatures in different contexts, also using examples from our own work. And then I will end by showing you other broader applications also to study human health. So really a disease of the genome. It's very much driven by mutations, and mutations accumulate during an individual's lifetime from very early on. And this is driven by both intrinsic mutational processes as well as environmental and lifestyle exposures. So throughout our lifetimes, we accumulate mutations, and most of these are really what we call passenger mutations. So they don't really affect the viability of our cells or induce any kind of disease. This is very much controlled by the immune system as well. But occasionally there will be a situation where what we call a mutator phenotype arises, whereby a certain mutation can lead to the development of diseases such as cancer. So once this mutator phenotype arises, then more and more carcinogen mutations will be accumulated in those cells, and that leads to full tumor development. And even more so, further mutational processes are acting upon those cells upon treatment. This is an ongoing process that we can study from very early on in healthy humans all the way to disease. And cancer is really the perfect example, because it really amplifies all this damage that is performed by different mutational processes, and it really fixes this damage in the genome. So this is why we are interested in it, and my group studies cancer evolution in its broadest sense from many different angles, but mutational processes are a key focus. So there's a variety of environmental as well as intrinsic factors that we are exposed to throughout our lifetimes. So there's aging, for instance, that can lead to mutations in our cells. But there's also environmental processes and risk factors such as smoking or alcohol, and they all damage the DNA of the cells in different ways. And the way to study this is by extracting this DNA from tumors and sequencing it. And by doing this, we try to understand what are the changes within the tumor genome that are arising that we don't find in a reference healthy individual. So what are those mutations that are present in the tumor cells that clearly have accumulated during an individual's lifetime and may have contributed to that disease. So when we do this, we find that cancer genomes are really very highly altered in many different ways. And here I'm just showing you one example of a full cancer genome. And this is from an esophageal cancer patient, but it is, I would say, quite representative of a variety of cancer genomes. And what you're seeing in the circular plot are the different chromosomes from 1 all the way to 22X, and Y is missing here. what is represented in the dots are the different somatic mutations that are accumulated in every single chromosome. And the arcs show you different rearrangements between different parts of chromosomes. So you can see that this genome is really highly altered. It's highly mutated. There's a lot of different segments that are broken from one chromosome and stitched into another. And this is quite representative of what we generally see in cancer. So we do see an accumulation of both single base substitutions that are not just randomly distributed throughout the genome, but they do follow certain patterns, and I'll be talking about that a lot in today's talk. But we also see other types of alterations, such as changes in the copy number of different segments in the genome, or inversions, translocations, different types of structural variation. Beyond this, we can see larger scale events as well. So there are cases where the entire genome is doubled, or cases where you have breakage fusion bridge cycles where certain chromosomes are broken and then fused together, and this leads to a lot of inversions and amplifications in the genome. There are regions that are hypermutated, meaning that they accumulate an unusually large number of mutations, and this process we call contagious. And there are also other catastrophic events that can happen, such as entire chromosomes, or at least chromosomal arms, breaking, and then the cell tries to repair this damage, but it doesn't really know how. So then all these broken pieces get stitched back together in a random fashion. And this is a very fascinating phenomenon called chromathripsis. So I will be mostly focusing on point mutations in today's talk, but it's important to be aware that this damage in the genome originally is really caused by different mutagens and different risk factors in the environment, as well as intrinsic risk factors that can damage the genome in such a way. So it's important to understand how mutational processes can actually cause such damage, and how we can use this to better understand the disease and also potentially treat it or diagnose it. So genomes of the same cancer can also vary widely. Here I'm showing you 6 different genomes from cancer patients. These are all esophageal cancer patients. And you can see that these are really quite strikingly different. You can have genomes that are really highly mutated and highly rearranged, or the opposite. There are some genomes that are quite quiet with very few rearrangements and few point mutations, if at all. So there's an extreme level of heterogeneity, which really complicates things in cancer. And this is why we keep on identifying new subtypes of different cancers. And this helps us better study this disease, which is not one disease, but it's a multitude of diseases. Now, just like footprints can act as evidence of historical events. So for instance, from this really beautiful book from Richard Deacon, you can see here footprints of dinosaurs. They're evidence that dinosaurs existed. Or a boot print of Neil Armstrong on the moon is evidence for humans having been there. Very similarly, we can think of mutational processes and what they do in the genome. So very similarly, we can see mutational footprints if we sequence genomes of different cells in cancer cells, and not only. So this is because different risk factors that are acting on these cells and are damaging the DNA, they do leave a footprint behind. Most of them, not all of them, but at least some of them do leave a footprint. And that means that the mutations that they induce in these genomes are not randomly distributed, but they are targeted at specific locations. So for instance, here on the left-hand side, I'm showing you a footprint of smoking. smoking tends to induce C2A substitutions, but not just randomly again in the genome, but in a very specific context. So most of the times when the adjacent bases are both Cs, so you have this nice trinucleotide pattern, which if you see in the genome, then you would say, okay, this is evidence that this cell or this tumor has been exposed to this patient has been exposed to smoking, and this is how the tumor may have formed, or at least smoking contributed to the development of that cancer. On the right-hand side, I'm showing you a different footprint. That is a footprint of aging, and this is C2T substitutions in a different context where the immediately adjacent 5' and 3' bases are A and G. So this is quite a distinct footprint, as I hope you can appreciate. Now we call these footprints mutational signatures, and they have been extensively studied, and a lot of the bases to studying this have been set up by the Mike Stratton group and Ludmila Alexandrov, who is continuing now in his own group along these lines. So these are very nicely cataloged in different databases, and they are continuing to be explored, and new mutational signatures are found all the time. Now, it turns out that the patterns are a little bit more complex than I showed you before, but not that much more. I hope that you can appreciate that these different patterns that I'm showing you that are linked to aging, smoking, defected DNA mismatch repair, and so on, that they look visually quite different. So for the human eye, they're quite easy to distinguish. Now, what are we exactly looking at here, or what are we representing? Well, we are looking at what is the probability that certain bases will be mutated if a certain risk factor is acting on those cells. So for instance, here, I'm showing you a signature of aging, of the aging process. Now, aging tends to induce C2T substitutions, like I showed you before. And particularly, there's a high probability of having C2T substitutions in specific contexts, as you see here, when the adjacent bases are certain specified ones, and pretty much zero probability that the aging would induce any other types of mutations in the genome. So this is quite a specific pattern, and we have such patterns that have been linked with a variety of risk factors. So as an individual develops throughout our lifetime, being exposed to different types of carcinogens and intrinsic processes that may damage the genome, we will accumulate such mutations, and they will, of course, induce different types of patterns in the genome. And all these patterns in the end come together to form the final genome when the individual is sequenced. So if this is a cancer individual, we will see actually quite a lot of these mutations. But and now comes the question, how do we actually infer these mutational signatures? Because when we sequence the individual, what is happening is that we have this final mutational portrait. So at any time point, if you take, for instance, a tumor sample, you will find all the mutations that have been accumulated in that tumor up to that point. So there will be a large array of base substitutions distributed in the genome. But of course, these substitutions have originated due to different mutational processes. So some of them happen throughout our life, such as aging. Some of them are intermittent. So for instance, smoking, maybe an individual was smoking only for a certain amount of time and then they quit, but they can be quite intense. Or intermittent mutational processes. For instance, acid reflux also damages the DNA of cells, and it is an intermittent process. So how do we actually go from this, trying to reconstruct from this final mutational portrait of the cancer to reconstruct the different processes that will have acted upon that genome and will have induced different types of footprints in the genome? So this is very nicely then illustrated in a mathematical problem, and I'm going to try and give you a very basic intuition for how this works. The methods that are applied, in a sense, are fairly straightforward, but they're very effective. But yeah, just to give you a basic intuition, what we want to infer are well. There are multiple things. So we have these different risk factors. Let's say we have 3 different risk factors that are acting upon a genome, and they tend to induce different types of substitution. So the red risk factors will induce mostly C2A substitutions. The blue risk factor will induce mostly C2T substitutions, and the green risk factor will induce mostly C2G substitutions, but also a little bit of C2T substitutions. So this is, sometimes we know exactly how the risk factor is working and what type of damage it induces in the cells. Sometimes we don't. So sometimes we need to infer this. But assuming that we know this, another thing we know is the actual final mutational count. So we can sequence a genome, and then we identify all the base substitutions in that genome. And then we can just count how many C2As do we have, how many C2Gs, how many C2Ts. So those are, this is the final mutational count. And in its most basic form, this is just a regression formula where we model the mutational count based on a combination of the different risk factor probabilities. And these probabilities are multiplied by the weight of the contribution of the specific risk factor. So if a risk factor contributed maybe 70% of the mutations in that genome, it would have a higher weight. If it contributed only 20% of the mutations, it would have a lower weight. So it's really these weights, or we call them exposures, that we are trying to infer generally. And we're really interested in this because this will tell us that a particular risk factor was very important for developing this disease, and others less so. And maybe other risk factors are not present at all. So then we know that that disease was not caused by that specific mutational process. So in its simplest form, this is very easy to solve because it's just a regression problem, of course. When it becomes harder to solve is when you don't know the P1, P2, and P3 probabilities. So in that case, what you actually have is usually a cohort of different genomes. You have the final mutational counts for all of those genomes. And then you're trying to write this equation as a product of the mutational probabilities of every single risk factor, as well as multiply by the exposures or the contributions of these risk factors to individual genomes. So you do know P, you do M, but you don't know P and E. So you are trying to infer both of them. Now, the good thing about this is that you have some constraints on the system. So the probabilities, being probabilities, have to be between 0 and 1. And also the exposure, so these are the contributions of the different risk factors to the genome. They are also in terms of percentages. So again, they have to be between 0 and 1. And this is a problem where non-negative matrix factorization therefore works very well. And this was first applied by Ludmila Alexandrov and Mike Stratton and Serena Nick-Zinal in the context of cancer. And it works well for a variety of problems. So if you have non-negative matrices that you are trying to write as a product of two other matrices, this works very nicely. And it works by basically trying to, you're randomly initializing the P and E matrices, and then you try to update them such that you get this product as close as possible to the M matrix. So this is kind of a brief overview of how the methodology works. Once you apply this, you can actually achieve what you meant to achieve, which is getting one cancer genome with all the catalog mutations, and then being able to say, okay, I have a risk factor like smoking, and this risk factor contributes 75% of the mutations in that genome. And I have another risk factor such as alcohol, and this contributes 10% of the mutations in the genome. And this is very useful for us because then we can start to better understand how those cancers developed. Now, like I said, these mutational signatures, they are not, or mutational patterns, they're not randomly distributed in the genome. We do see these local effects where the context, the 5' and 3' bases matter a lot. But there are also other properties that influence these signatures. And this have already been cataloged some years ago. So we know that there are certain key processes that happen during the cell cycle, and you know, the cancer cells, they divide all the time, so you have these processes happening all the time, or yes, during protein production, transcription, translation, that really affect the way mutations will be distributed in the genome, will be accumulated, and also will be repaired. So in terms of transcription, it turns out that depending on which strand the mutation is accumulated, this will be easier or less easily repaired. So there are certain biases in terms of how mutations are accumulated on different strands and how they will be repaired. Secondly, during DNA replication, there's also some biases happening. So depending on whether you are on the leading or the lagging strand, again, mutations, certain types of mutational processes will induce mutations that are preferentially accumulated on one of these two strands. And also the efficiency of repair, again, is different on the leading and on the lagging strand. So having analyzed these types of effects, several groups in the past have come up with this result that certain mutational processes really show a very strong transcriptional or replication strand bias. And not all of them show this, but one particularly interesting process for this is this process called signature 17, and we call it that because we don't really know yet what the etiology of that process is. I will come back to it, but yeah, it does have a very strong both transcriptional and replication time bias. So do keep in mind signature 17 because I will come back to it. Furthermore, it's not only what's happening at the different phases of the cell cycle, but also where mutations are generally falling within the genome. So, for instance, other groups have shown that binding at transcription factor binding sites does tend to show a bias. So there are certain mutations that are more strongly accumulated exactly at the transcription factor binding site, and then they would not be kept in the vicinity of the binding site. Furthermore, the nucleosome periodicity also seems to play a role. So certain mutational processes, such as signature 17, again, I'm coming back to that, are displaying this preference that is very much influenced by the nucleosomal periodicity. So they tend to be accumulated in a very periodic fashion, with this periodic frequency that can be written as a function of the distance from the diet. So these kinds of processes will be very much impacted by the chromatin architecture. And there's still a lot we don't understand in terms of how the genome is organized, especially in 3D, and how this might impact the accumulation of mutations by different processes within the cells. So before I will now, before I discuss my own work, I want to give you some other examples of how these mutational signatures can be applied in a variety of scenarios. So hopefully you're already getting the feeling that we can study them to get an understanding of how cancer develops. But what about beyond that? Are they useful beyond just, fundamental knowledge of the basic biology of cancer? I would argue that yes, they are starting to become useful in a variety of scenarios. And one example I'd like to give you is that of chemotherapy, which is one of the most mainstream therapies given in cancer. So this is a standard therapy. Most of the cancer patients will get it. And it's a very generic therapy that targets cells that are proliferating in the body. Now, Nuria Lopez's Bigas's group have studied the effects of different therapies on the genome of cancer cells. And what they found out was that upon chemotherapy, you tend to see an accumulation of T2G substitution. So they identify the mutational signature that is associated with the effect of a particular chemotherapy, in this case, 5-FU. And this is something that recurs, appears recurrently across many different cohorts. So it's suggesting to us that it's actually something that people weren't aware of before. So giving certain drugs to patients, we knew that it can lead to curing these patients, or it can cause resistance through other mutational mechanisms. But up to the point of that study, it wasn't clear whether the drugs had more general, broader effects in the genome. And what this study showed was that you can have this mutational process induced by chemotherapy that leaves a very specific footprint in the genome, this of T2G substitutions. Now, this has implications in particular for childhood cancers, because childhood cancers, some of them at least are treatable. So quite often children will get chemotherapy, but this means that this might have longer term effects. If they receive chemotherapy and are cured, still they will have accumulated these mutations in the cells that are caused by chemotherapy, and they might have longer term effects. So in certain cases, one might want to see if there are other therapies available that would not have such long-term effects. Another very convincing example that I want to give comes from mostly from breast cancer. Now, breast cancer is quite a well-studied cancer, and it has also quite a strong inherited component. So quite a few women might present with a germline mutation and the genes BRCA1 or BRCA2, and this confers them an increased risk for breast cancer. Now, why is this so? This is because these genes, BRCA1 and BRCA2, they are involved in double-strand break repair. So if you have double-strand break repair, double-strand breaks in the genome, these genes will come and they will trigger the processes that are needed to repair these breaks. Now, if you are missing one copy of these genes, then of course the repair process will be less effective. But the cells still can survive. So what happens in cancer is that quite often, if you have a BRCA1 or 2 mutation, then indeed this process is impaired. So the cells have a sensitivity for double-strand breaks damage. And what one can do then is intervene with a PARP inhibitor. So a PARP is another gene acting on this pathway. So you basically hit that gene, and this leads to the collapse of the whole double-strand break repair pathway. So then the cells are not able to repair the double-strand breaks anymore, and that so it leads to synthetic lethality, and you can kill the cancer cells. So these PARP inhibitors have actually been proven to be really successful in a variety of contexts where you have this type of deficiency, which is called homologous recombination deficiency. Now, what another group, Serena Nixzinal's group, did a few years ago was to study a large cohort of breast cancer patients. And we know that there are certain patients that display these classical homologous recombination repair deficiencies that are linked to mutations in BRCA1 or 2. And that's represented by these colored yellow, purple segments of this pie chart. Now, there's a considerable fraction of patients that don't exhibit these alterations. So the ones that do, they can benefit from PARP inhibitors. But what about the rest of the other patients? Well, it turns out that there were quite a few patients who didn't have the classical mutations in BRCA1 or 2, but they, when analyzing their genomes, the authors found that there was this prevalent mutational signature that is associated with homologous recombination deficiency. So this is the signature shown here. And this suggested that that pathway might be impaired or sensitized in the absence of BRCA1 or 2 mutations. So this has led to the authors to stipulate that these fraction of patients that have this mutational signature of homologous recombination deficiency could also be sensitive to PARP inhibition, so they could also benefit from this treatment. And now there are ongoing clinical trials that seem to be promising in this respect. So this is another way to illustrate that mutational signature certain cases could also inform treatment. So before I go into our own work on mutational signatures, I would like to take a quick break and ask if there are any questions up to this point.
Actually, yes, there are two questions from Anubrata. So she is asking if there is any way to know if a cell is tipped to acquire a mutator phenotype.
Yeah, that's a very good question. It's also quite difficult to answer, I think. Yes, there's still a lot we don't understand about these mutational processes and how they are accumulated in the cells. In principle, well, I'm trying to think of studies that would kind of try to link towards an answer for this. I can tell you there have been some studies in cell lines and where you expose cells to different carcinogens, and then you can observe whether they're accumulating a certain mutational signature or not. And that works better in certain cell lines rather than in others. So that would be an indication that, yes, maybe the specific tissue of the cell line or just something about it that cell makes it more prone to accumulate mutations from that process. But that said, I think it's mostly depending on where different carcinogens are acting, where they're accessible within the body. If we think about cancer, for instance, we will see a lot of alcohol-linked mutations in the liver. Or we will see UV light-linked mutations in the skin and in skin cancer, but we won't see them that much, for instance, in the esophagus that is internal and is not exposed to UV light. So I couldn't really answer this very well because this is still very much ongoing research, but I would say it's probably a little bit more to do with whether the cells are actually exposed to a certain risk factor and how much for how long they're exposed to it, rather than intrinsic properties of the cells. But that said, I'm probably wrong. We'll probably find out in the future that it has to do a lot more with the cell makeup. But if you think about it, in principle, in the healthy body, the genome is all the same, right? Of course, there's different expression and different cell types, but the genome, in principle, it should be identical in all the cells in the body. So it should be equally prone, I would think, to to accumulating such mutations. But once it's accumulated certain mutations, it probably is less prone to accumulating others. So yeah, there are dependencies definitely there.
Thank you, Maria. And there is another question also from Anubrata. How are individual risk factor weights determined? So the weights for individual risk factor.
Yeah, so that's with this matrix factorization process that I mentioned before. It depends. So if you already know the risk factors that are acting on that genome, because they have been described, like in the common cancers, we know quite a lot about which risk factors are acting on those genomes, and we know what kind of pattern they induce, then it's a simple regression problem. So this is very easily inferred. Otherwise, if you have a new cohort that is maybe a cancer type that has not been studied extensively, then you want to uncover de novo mutational processes as well as their weights and contributions in the genome. And in that case, you apply matrix factorization, non-negative matrix factorization. And yeah, it's about initializing these matrices with random values between 0 and 1, and then multiplying them and updating them up to the point that it becomes as close as possible to the actual observed data in the genome. So you try to minimize the error. But you can find a lot more details on that on the expert who developed this methodology for mutational signatures, who is Ludmila Alexandrov. So if you read some of his papers, you will be able to get many more details on that.
Thank you, Maria.
Great. Okay, so if there are no more questions, then I'll go on and tell you a bit about how we've been trying to study mutational processes in the context of esophageal adenocarcinoma. Esophageal adenocarcinoma is quite an interesting cancer to study. So it's cancer of the esophagus. There are two cancers in the esophagus, esophageal squamous cell carcinoma and adenocarcinoma. And adenocarcinoma tends to occur closer to the gastroesophageal junction. So it's lower down the esophagus. It has a very poor prognosis. Generally, only 5% of these individuals, 15% of these individuals go on to live for a period of 5 years or longer. And it's much more prevalent in the Western populations. Now, the reason why it's quite interesting and quite well fit to study cancer evolution because it develops through a precancerous stage. So we know that there is this precancerous condition called Barrett's esophagus. And this is usually caused by acid reflux from the stomach that comes and basically burns the lining of the esophagus and induces a lot of mutations there. And this is a benign condition. It can be a benign condition, but 3% of such individuals that suffer from Barrett's esophagus will go on to develop esophageal cancer. So it's not always that they go on to develop esophageal cancer, but sometimes they do, and this is why it's quite interesting and intriguing to study. So once the cancer has developed, it goes through a stage of dysplasia, and then you have the full-blown tumor there. And the tumors metastasize quite rapidly. From other studies in the group, we found that they actually tend to metastasize years before they would actually be discovered in other parts of the body. This also relates to the very poor prognosis that these patients have. But as you see here, we have a whole timeline of cancer evolution that we can capture if we sequence these individual stages. And this is what we've been trying to do in our work. So firstly, I'd like to mention an older study that we did on a smaller cohort of 109 esophageal adenocarcinoma patients. So we took their tumors, we performed whole genome sequencing on them, and then we identified these mutational signatures that are acting in these genomes using the methodology that I just described before. This was nice because it gave us an overview of what are the main mutational processes that lead to the development of this cancer. So these include both intrinsic processes, well, I would say mainly intrinsic intrinsic processes. And yeah, we see some very prevalent processes such as aging. We see aging pretty much in all cancers. ApoBec activity, this is a family of enzymes that I'll talk about later. We also see a lot of mutations caused by different DNA damage repair deficiencies. Some sort of extrinsic processes linked to oxidative stress. And also this signature of T2G and T2C substitutions, that is called signature 17. So if you remember, I told you to keep it in mind because I'm going to come back to it. And this is really a very important signature in esophageal adenocarcinoma because it is the most prevalent mutational process we observe in these tumors. However, we don't really know what the underlying cause of this is. So we don't know what is causing this mutational pattern. We know it's very prevalent. There are different hypotheses. It may have to do with some inflammatory processes in the esophagus. It's really not known and very difficult to prove. But there are a lot of groups that are trying to understand this. So what we found was, like I said, the signature 17 process is the most dominant within these patients. It's found in more than 50% of the patients. But we were able also to group and classify these patients based on these mutational signatures. So we identified three main subgroups. One is the one that's dominated by this very strong mutational process. Another one that is linked with DNA damage repair impairment, and another one where aging and oxidative stress are the main factors that are causing the development of the disease. Now, this was useful for us because we could not only understand how these different patients, what the course of development of the cancer was in these different patients, but we could also reason about what was happening in the broader context and the pathway deregulation happening in the subtypes of esophageal cancer. So it turned out that the patients that had this signature 17, they had a really high mutational burden, and they seemed to have a stronger immune response. So we were actually able to propose therapies that are not really used as standard in this cancer for these subgroups of patients. So this one, we thought they might benefit from checkpoint blockade. The DNA damage repair impaired subgroup, that it has a similar homologous recombination deficiency like I discussed before. So these patients could potentially benefit from PARP inhibitors, and the rest of the patients would just go with the standard of care. So these are hypotheses that we are currently testing in a variety of scenarios. But this gave us, a first overview of the main mutational processes acting in these genomes. Now, this is still quite a static picture because we're looking only at the final point when we sequence the tumor. And it's telling us, okay, there are these mutational processes. But in reality, things are a little bit more complicated because cancer is an evolutionary process. So it's not that you have a constant accumulation of mutations. So you start with the healthy cells, and then you start to accumulate mutations that will expand within the tumor. So you will see different clones that are forming, and the clones that are fitter, that accumulated those mutations that confer an increase in fitness, they will expand within the tumor. Other subclones that are less successful will eventually die out. So there's this ongoing dynamic process that is happening, and that is, of course, very much influenced by different mutational processes acting at different time points, but we're yet not modeling this properly. Now, there is a way to formalize this through phylogenetic trees. We can place every mutation on branches of the tree, and this kind of gives us an overview of how these mutations have led to the development of the disease, and which branches were more successful than others. But how do we link this to mutational processes? So we have to think of it as, because it's a dynamic process, it may be that certain carcinogens, yes, they act all the time, like aging. But other carcinogens might only be acting at certain time points during tumor development. So you will have some that are acting earlier on, some that are acting later on. So how do these dynamics impact cancer evolution? Well, we try to reason how we could analyze this. And we still are limited. Ideally, in an ideal scenario, you want to take many samples at different time points in cancer evolution, and then you can see what's going on at every time point. And that's a very clear picture. But of course, we don't have the luxury to have this. Usually, we have a single sample from the patient that was taken at the time of surgery. That sample is sequenced. So we really just have one time point. However, within that time point, we can infer some time information from this. And this is because if we think about how we are aligning reads to the genome, there is an additional information that is embedded there. So for instance, when we take the sequencing data, we start to align the reads, we might find that in a region, 2 of the 5 reads that were aligning in that region are mutated, and the other 3 reads contain just the reference nucleotide at that position. So if this happens, that actually suggests that if we had 5 cells in our tumor, 2 of the cells would be mutated, would have a mutation, and the other 3 cells wouldn't. So this is a very rough approximation of the reality, but this is kind of the intuition, and this is how we reason about things. So if we actually plot the distribution of these mutations across the genomes and their frequencies across the genomes, we start to see a picture like this, where you see multiple peaks in this histogram. And the peak that is closest to 100%, the A peak here, usually suggests that these mutations are found in most of the cells within that tumor. So they are actually clonally present. And that would mean that they have very likely been accumulated very early on during cancer evolution, because they are present in all the cells, so they have been kept for a long time. And then you see some other peaks, B, C, and D here. So B and C are sort of intermediate frequencies. These are 2 subpopulations of cells. We call them subclones that contain some mutations. So generally within tumors, you see this very heterogeneous population. So they would contain specific mutations. And then you have the D subclone, where the mutations are very infrequent. So some of these would actually be neutral mutations, some passenger mutations that don't really have a fitness effect. So we can then start to plot this on a tree, and then we would have the A mutations, so from the subclonal population that is at the top of the hierarchy. So that's the initial clone that initiated the tumor, and then the mutations in that clone were actually kept throughout cancer evolution and all the other cells, because they were so beneficial to start with. And then the cells, different parts of the tumor are exposed to different stresses, and they will need to react to those stresses and accumulate different mutations afterwards, and that leads to further subclonal development. So the way we formally construct this is through a variety of different methods. Bayesian Dirichlet processes are one commonly used one that allows us to place these mutations into subclones. And this is pretty much the framework we used in order to assign mutations into 2 categories, clonal and subclonal. And then we can see which clonal mutations are linked to which mutational signatures or processes, and which ones are subclonal. And we believe that the clonal mutations, again, they appeared earlier in cancer evolution, and the subclonal ones later in cancer evolution, although this is also a simplification and it's a limitation of our approach. So having established this framework, what did we then go and do? Well, we have a very nice study set up where we are able to sample from different stages during esophageal cancer evolution. So we have Barrett's esophagus samples, primary tumors and metastases, quite a few of them, and we've sequenced them. And then we identify mutations in all these different samples. We look at their clonality and timing. We reconstruct mutational signatures, and then we identify evolutionary bottlenecks where these signatures and these mutational processes are changing and study that. So first of all, by studying this larger cohort of cancers, and I should mention that this is very much a collaboration, and this study was initiated in Rebecca Fitzgerald's lab, and Sujat was the PhD student who studied these processes, and Rebecca and I co-supervised him. Right, so what did we find? First of all, we expanded the landscape of mutational processes that are happening in esophageal cancer. We see now 14 mutational processes. We still see the key ones, such as the signature 17 process. We see processes related to apobec or oxidative damage, but also some other processes. Base excision repair, processes linked to chemotherapy treatment, and also mismatch repair deficiencies. So it's a broader landscape, more processes, because we have more power to detect such mutational signatures in the genome. We also had matched cases where we had the Barrett's esophagus, and we also had the primary tumor from the same patient. So we actually had the evolution of that disease in that patient. And then we were able to tell that certain mutational processes are increasing in intensity from Barrett's esophagus to primary tumors, and others are decreasing. And I won't go into details, but I would like to highlight the SBS2 process, which is linked to apobec activity, and I'll come back to that, but it seems to increase in the primary tumors. Now, how can we study this more formally? So like I said, we do want to identify these evolutionary bottlenecks where mutational signatures are changing. And what I mean is that first you have a certain mutational processes represented by these different colored lines, and they all contribute to a certain extent mutations in the genome. So the green one is the most intense here, and it's contributing to 20% of the mutations. Now, then at the bottleneck, something changes in the system, and the green process becomes a bit more intense, but other processes really drastically decrease in intensity, such as this magenta process that goes from 15% to something like 7%. Of the mutations. So it's these kinds of changes that we're trying to capture, and we call these bottlenecks. And we find that there are such bottlenecks where the mutational signature dynamics is changing in 51% of the cases in our tumors that we study. And what is happening at these bottlenecks? Well, we see quite a few changes, not many drastic changes in most mutational processes, but we do see quite strong changes in the signature 17 process. So what I'm plotting here for every single mutational signature, I'm plotting you the change, the direction of change of that process at the bottleneck. So if we're above 0, it means there's an increase in the intensity of that process in the genome. If it's below 0, there's a decrease in intensity later on in cancer evolution. So this is when the bottleneck is happening. And what we see predominantly with signature 17, both there's 2 of them, A and B, we see that both of them tend to decrease at the subclone or at the evolutionary bottleneck. So that means they become less intense during cancer evolution at a later time point. And we still don't know why this is happening, but it's quite interesting. It's suggesting that it may be replaced by other processes or maybe mediated by inflammatory processes or drug treatment. Now we do see that signature 17 also displays hallmarks of genomic instability. So samples that have been exposed to this process have an increased ploidy, an increased chromosomal instability, here shown in red, also increased processes that have to do with telomere maintenance, DNA damage repair, cell cycle control, and angiogenesis. So it does look like these tumors are really much more altered, much more unstable in relation to this process. So this, of course, suggests that these tumors might be more successful in progressing later on, but then there's this evolutionary bottleneck that is happening, and it might slow down this progression at some point. Beyond signature 17, what are the other processes that are contributing? There's a variety of them, and here it's a really complex landscape that I won't even try to explain because it's not really possible to explain it in this way. But very briefly, I want to show you how we've been trying to model this. The way we've been trying to model this is by looking at whether we could, we reasoned that there's different mutational processes. They're acting in different combinations in different patients, but also at different stages of the disease. So is there a way in which we might determine that certain mutational processes are more important for the transition from Barrett's esophagus to esophageal cancer, or from the primary tumor to the metastasis? Are there certain processes that act primarily in one stage? So to model this, we asked, can we model the cancer stage based on the combination of mutational processes while taking into account the clonality of the mutations and their timing as well? So to do this, we use two approaches. We use a random forest approach and a gradient boost approach. And again, we're trying to predict the cancer stage, whether it's Barrett's esophagus or primary tumor, based on the combination of mutational processes. And we do quite well, as you can see here, in predicting whether the sample is a primary tumor or a Barrett's esophagus sample, just based on mutational signature information, which is quite remarkable. Here are the mutational signatures that are contributing most to this prediction. This is a Shapley plot that is showing you the individual contributions of the different signatures. The signatures are ranked according to their importance in the model. And the different dots here correspond to different samples. The color of the dots correspond to the prevalence of that mutational process in the sample. So here we're looking at distinguishing Barrett's esophagus from primary tumor on the right, Barrett's esophagus on the left. You can see that signature 2 is higher, more prevalent in primary tumors, whereas and signature 40 as well, whereas signature 1 is more prevalent in Barrett's esophagus. We also see that clonality here in blue. Blue means clonal, yellow means subclonal. So there's more of clonal mutations in Barrett's esophagus, which we expect because it's an earlier stage of the disease. Also, the timing of the mutations is earlier. It's more linked to Barrett's esophagus. Again, early mutations would be expected in Barrett's esophagus. So that's reassuring. But now I want to focus on the main mutational process that we obtained here, which is SBS2. This seems to be the most important one in distinguishing the two stages, and it seems to be more prevalent in primary tumors. Now, SBS2 is linked with ApoBec mutagenesis. ApoBec is a family of enzymes that is involved also in viral processes, in viral infection, and it induces cytidine deamination in the genome. It's quite prevalent in cancer. We definitely see it in our cohort, and it's also been shown to be linked with certain mutational hotspots. So in the presence of ApoBec mutagenesis, you tend to observe these mutational hotspots in cancer genes such as PIK3CA or KRAS. Now, we wanted to have a look at this as well. Could this be linked to certain driver genes of esophageal cancer? And for this, we used a method that is called DNDS. It's used in population genetics, and that basically allows us to assess which genes accumulate an unusual amount of missense and nonsense mutations compared to synonymous mutations. So in other words, which are the genes that are positively selected in cancer? So when we do this, this is what we're seeing as positively selected genes in Barrett's esophagus, primary tumors, and both of them. So the blue genes are the ones that are positively selected in both Barrett's esophagus and primary tumors. The red genes that are positively selected only in primary tumors, and yellow genes only in Barrett's esophagus. And here's when we compare metastasis to primary tumors. Again, the red genes are the ones that are specifically positively selected in primary tumors, but non-metastasis. And what we see here is that in primary tumors, we have KRAS and PIK3CA specifically selected in that cancer stage, and not in Barrett's or not in metastasis. And the Appleback process was also very strongly linked to primary tumor stage, which is suggesting to us that maybe Appleback is appearing at the stage of transformation from Barrett's to primary tumor, to primary cancer. And it does so by enabling these cancer drivers that seem to be important for this stage. Right. So with this, I would like to summarize by saying that hopefully you get an idea of how we can use mutational signatures and study their dynamics in order to get more understanding of how this cancer evolves. And potentially some of these insights, because some of these mutational signatures tend to be quite state specific, they could in the future be used as marker of this stage in cancer. But there's a lot we still don't know about mutational signatures. And so in the last minute, now I will just very briefly show you that really, well, there's a lot that's left to explore. So first of all, the mutational signature catalog is continuously expanding, where we went from something like 30 signatures to 80, and now there's more than 100 signatures that have been described, and this will getting bigger and bigger. There are not only mutational single base substitution signatures, but also copy number and chromosomal instability signatures have been described actually very recently. 2 papers published in Nature a couple of weeks ago very excitingly described these larger scale processes that contribute to cancer development. We're starting to understand a bit more about how different carcinogens might induce mutations in the genome by studying these processes in vitro, in cell lines, and also in mice. So we understand that certain carcinogens do induce patterns of mutations and certain ones don't. And also recent studies have shown that mutations are not only present in cancer cells, but we have mutations in our body all the time. So seminal work from the Marty Corena and Mike Stratton groups and Peter Campbell's groups have shown that there are mutations in healthy tissue. They've sequenced healthy individuals, and they've shown a myriad of mutations that you can see represented here with different circles depicting different clonal mutations. And these often hit genes that are involved in cancer, but the patients are completely healthy. So this is suggesting that we can start to study these mutational processes very early on, and hopefully also detect cancer earlier at some point if you can understand how these mutational processes are shaping the cancer genomes. We see mutational processes prevalent in other disease conditions such as cirrhosis, and here you can see spatial effects of mutational processes in different parts of the the liver. So people are starting to study this. And there is the Mutagraphs effort that was funded by a CRUK grant challenge. And this one is aiming to study mutational signatures, not only from Western populations, but throughout the globe, taking into account and recording all the different behaviors and exposures that different people in different parts of the world may have, so that we can better understand how different risk factors lead to the development of cancer. And mutational processes are not only present in human, they're present across mammal species. Again, very recent work published in Nature showed that these processes can be quite similar between different species, but certain processes accumulate at different rates in mouse and naked mole rat, for instance, compared to human. So this is also showing there are some broader evolutionary effects in the way we accumulate mutations due to different processes. So this is just showing us that mutational processes, there's a lot to discover there, but there is potential to use them both for diagnosis, for treatment of disease, but also for the fundamental understanding of different evolutionary processes in cancer, but also, you know, in the broader evolution between species. And with this, I'd like to end by acknowledging Rebecca Fitzgerald and all the group, Sujat, for the work that I've presented in the context of esophageal cancer. And thank you for your attention.
Thank you very much, Maria, for your great talk on cancer mutations. I spotted, to be honest, a couple of things that I would like to read or to focus on. And I also liked kind of like towards the end this part with I'm specifically interested in cross species comparison, so looking to these studies also in rats models would be also nice, and to other types of cancers. I actually was thinking to how someone could, for example, reproduce, let's say, your work. It's great work, but I'm just saying in another type of cancer. So, of course, there is the biomedicine input that is needed, but I think some parts, at least at computation level, can be inspiring. So,
yeah, so I mean, we generally try to document everything we're doing quite as well as we can. I should say, you know, none of this is really entirely novel. There's a lot of people that are using similar approaches in the field, so I have to give a lot of credit to everyone who set the basis for mutational signature analysis. There are many different groups. I didn't mention them, and in fact there are multiple approaches to detecting mutational signatures. We're also currently working on different types of approaches for detecting such signatures. But yes, I think, you know, in terms of applying this to a different cancer, especially the part where we look at the dynamics of these processes, yes, this is completely applicable to other cancer types. What we'll see, of course, depends very much on the cancer type, but the kind of approach we're taking, yeah, it can be easily applied to other cancers. And I think this is where the field is going as well, trying to incorporate more information, you know, about the timing of these signatures, as well as going beyond just mutational signatures, but incorporating also other marks, genome, like transcriptional information, chromatin organization, and so on. So there's still a lot to be explored, and I think we are definitely looking a little bit in a silo, you know, just very focused on the mutations, but there's definitely a lot of other processes, larger scale rearrangements that need to be taken into account. So I think, you know, that our approach can definitely be applied, but there's also a lot of scope for expansion.
Exactly. I was thinking actually during your talk to the epigenetic events or interdependencies between epigenetic and genetic events. But I was also thinking to the rare cancers. So I'm not sure. I saw the cohort is quite well represented in your studies, like more than 100 in one part. For rare cancer, usually they work with less than that.
Yeah.
And another thing I was also pointing, because some interest we have in gender differences for the patients, and how this affects the patients, or I'm not sure if you studied that, look to, or yes, just saying.
Yeah, I think that's very interesting. And yeah, again, very, very new research that is starting to be done. So there have been studies from several groups showing that mutational signatures do differ by sex. So yeah, you do see a different distribution in men and women, and you also see a different distribution and based on the ethnicity. So yes, that is starting to be studied. In the context of cancer, still the sampling is so that we mostly have samples from Western populations, European and Americans. But yes, but this is changing now. So hopefully we'll have a more representative sample across the globe. With respect to your points about epigenetics, absolutely, that's a really good point. I think well, people have been trying to develop methylation signatures for a while, but they haven't somehow been as successful for some reason. But I think the problem is, again, integrating. There's work from Moritz Gerstung's group that is actually trying to integrate more than beyond mutational signatures, integrate different layers, so methylation, expression, and so on. The more you integrate, I think the trade-off there is that you lose interpretability. You get very, very complex signatures afterwards, and they're much harder to interpret. So this is why the field currently is still sticking mostly focusing on mutational signatures and not looking at other data types. But definitely this is upcoming, and I think there's potential for deep learning approaches in this context as well that will not be more interpretable, but maybe at least will give us easier ways to link to patient outcomes. And the rare cancers are indeed a difficult and particular case. So for rare cancers, the good thing is that initially it used to be that you can just infer mutational signatures only if you have a cohort of patients and it needed to be large enough. But newer methods are able to identify mutational signatures in individual patients, so in a single sample. It doesn't depend on having a cohort. So that's good for rare cancers, because there we don't really have a lot of power. Of course, for rare cancers, I think we're more limited in getting a good overview of unless it's a very homogeneous disease of the different ways in which that disease can develop. But more sequencing data is accumulated from all different types of cancer, so we'll be able to better study those in the future as well.
Yes, I think a special case is also for pediatric cancer, so where data basically is very limited or kind of like difficult to. And very important, I find also the fact that you mentioned mutations are present also in healthy individuals. So none kind of like escapes, depends now how they react, how they function and so on. So yeah.
Yeah, I think I mean, a big component of this is the immune system. Have to understand more about how that works.
Okay.
But clearly, you know, there's all these clones in healthy individuals that are really, by definition, they should be cancer clones. They should lead to cancer. It's all these classical genes, NOTCH1, P53 sometimes. So you do see them, the cells are mutated, but still the individuals don't develop cancer. And that's probably very much an interplay between, I don't know, something intrinsic that we don't understand, as well as how these clones are detected by the microenvironment and kept in check. So yeah, there's a lot of excitement in this area, and we're still, you know, at the very basis of exploring this in healthy individuals. But I think that's very exciting.
And we have another question from Yuri. So are mutational signature going to be relevant to our diseases of old age, example, neurodegenerative disorders and so on?
Possibly, yes. I think it's just we we haven't sequenced enough to really tell. I wouldn't want to comment too much on that because that's really outside my area of expertise. I do know there's a lot of efforts put into RNA sequencing in neurodegenerative disorders because there's a lot of changes that are happening in this respect. But I would imagine, because we see it in healthy tissue, we might see specific changes there as well. They won't be at the scale that you see in cancer, that's for sure. So they might be harder to detect. It will need to involve deeper sequencing. But yes, I think that's very possible. In fact, now there's efforts from the Sanger Institute looking at all sorts of other conditions, like I showed an example of cirrhosis, where it really looks like, or inflammatory bowel disease is another one where we also see these mutational processes very prevalent. So I think, yes, I'm not personally aware of research in this area in neurodegenerative disorders, but I think it's entirely possible. Yeah.
Yes. I tend to believe quite in the same lines, but yes. Thank you very much.
I guess, yes, it depends. If we're talking about the brain-related disorders, maybe the brain is a little bit more protected than other organs from external factors, but there's still internal processes. So yeah, definitely, I think.